Hi, in this video, you will see my group and I conducting experiment on cell staining. So let's begin with the introduction of the light microscope. As we know, microscope is used to see objects that are too small to be seen by the naked eye. As you can see on the screen right now, is a light microscope available in our laboratory. This is the main stage of the microscope. This is where we will put our specimen to be observed under the microscope. The most important part in the microscope is the objective lenses. There are 4 of them altogether. Next is the eyepiece. This is where we will observe our specimen under the light microscope. We can use the coarse focus and fine focus to adjust our lenses focus so the specimen can be seen clearly. The carrying handle is used to carry the microscope without breaking any parts of it. The next component of the microscope is the illuminator. The illuminator emits the light as the power switch button is turned on. The light intensity also can be controlled. The last component of the microscope is the stage control. You can either move the stage up and down or right and left. This will help you to make sure your specimen is located directly above the light so we can observe the specimen. Let's begin our first experiment to observe the animal cell. First, place 1 drop of methylene blue in the center of the microscope's light. Then, use a toothpick to scrape the inside of your cheek. Smear and mix the cells in this methylene blue solution. Put 1/8 of a cover slip to one side of stain and slowly lower the cover slip. Touch edges of cover slip with paper towel to remove excess stain. Now, let's observe the specimen under the light microscope starting from 4 times of magnification followed by 10 and 40 times of magnification. Now, let's move to the next experiment, which is to observe the plant cell under the light microscope. We use Allium SP, or in other name is called onion, for the experiment. The first step is to cut a small section of an onion. Use a forceps to peel off the thin transparent epidermis of an onion. Place the onion epidermal in the center of a microscope slide carefully. Now place few drops of iodine stain on the epidermis. Same as the animal cell, put 1/8 of a cover slip to one side of stain and slowly lower the cover slip as shown on the video. Then, touch edges of cover slip with paper towel to remove excess stain. Now, observe the specimen under the light microscope starting from 4 times of magnification followed by 10 and 40 times of magnification. For the last part of the experiment, we had observed the prokaryotic cell under the microscope. As we know, prokaryotic cells are much more smaller than animal cell and plant cell, so we had to observe the prokaryotic cell a bit differently from the animal cell and plant cell. Now, place 1 drop of immersion oil on the cover slip of the specimen slide. Swing in the 100 times objective lens and make sure the tip of the lens should now is in contact with the oil. Adjust sharpness and clarity using the fine focus knob.